Hi, and welcome to the audio slides of our paper. This paper was published in May 2016. I am Martin Oliver Steinhauser, the corresponding author of this publication. In our research, described in the paper, we conducted a comprehensive study of the self-assembly of lipids in a solvent. One of the major results, among many other things, is a deeper understanding of the dynamics of lipid self-assembly. More concretely, we found that the gradient of the second-order Legendre polynomials is strongly correlated with major orientational changes of lipids during aggregation. The relevance of our research is connected with the fact that we have been exploring experimentally the possibility to destroy tumor cells by shock waves which are generated by laser ablation. The destruction of tumor cells using laser-generated shock waves might open up new therapeutic forms of non- or minimal invasive medical treatments of tumor cells and tumor tissue. Our simulation study presented here provides a realistic computational model for the simulation of such cellular structures, which is needed to be able to simulate the effect of shock waves on the molecular structure of cells, for example, their plasma membrane or their cytoskeleton. This slide shows our coarse-grained, simplified particle model of a typical lipid molecule. The different chemical groups which build the hydrophilic head, displayed in red here, along with the hydrophobic tails, displayed in yellow, are merged into a so-called bead spring model, which, in essence, consists of particles connected by entropic springs. In experiments, you have to deal with the full complexity of the real system, the eukaryotic cell as seen on the left. In our endeavor to develop a computational mechanical model of a full eukaryotic cell, we focus here on presenting a comprehensive computational study of bilayer membrane structures. For performing the simulations presented in this study, we used the molecular dynamics simulation software suite MD-Cube, which was developed and written by Martin Steinhauser. We discovered that whenever a major change in the self-assembly of lipids occurs, this can be seen very well in the steep gradient of the order parameter. On the left, one can nicely see the gradual self-assembly of all lipids into one large bilayer membrane. On the right, these major changes in orientation are reflected in the fluctuations of the order parameter. Here we show the pair correlation function of the final membrane structures for different temperatures. The lower the temperature, the more distinct peaks, even at larger distances, can be seen, indicating long-range, crystal-like order. For higher temperatures, there is ever less structure, and only the first and second neighbor peak can be seen for the highest temperatures, indicating a gas or liquid-like state. Depending on the choice of particle interactions and temperature, we can distinguish four different distinct phases of our aggregated lipids. There's a gas phase, a fluid phase, a crystalline phase, and a phase where closed vesicles are formed. These four phases are displayed in different colors, and sample simulation snapshots of typical structures are provided here. Such a comprehensive analysis has not been done before in other computational studies. In conclusion, we have contributed considerably to an improved understanding of the molecular self-assembly of lipids. We've calculated a complete phase diagram of our new lipid model, and we have investigated the static and dynamic properties of bilayer structures and of vesicles. Our research, presented in this paper, constitutes an important first step towards modeling the mechanical features of a full eukaryotic cell. In future work, which is currently in progress, we will expose our bilayer membranes to shock waves. We will also combine our aggregated coarse-grained membranes with a polymer network to model the mechanics of a full cellular structure, including the cytoskeleton and the plasma membrane. This research will contribute to a better understanding of the effects that shock waves have on the mechanical stability of biological cells.